Okay. So, hello and good evening, everyone, to the extra live class session or the special live class session on the course Fundamentals of Protein Chemistry, which is being taken by Professor Sagota Dasgupta of IIT Kharagpur. So, today basically is the final session where we would be discussing a few things. Like if you guys have any doubts or something, we would be doing that. And also there is a portion that I wanted to complete, like the protein macromolecule interaction, that was also a part of this thing. And so if we go in this manner where let us try to maybe figure out or jot down what we had done so far. So we started with amino acids, the types of amino acids, where we discussed on the characteristics, the physical properties of amino acids, and so on. Then we talked all on things like the proteins and the different properties of proteins, the different types of proteins that are present in the different, and the function, the structure of these proteins and all. Then the peptide bonds, the role of water in this formation of these peptide bonds, followed by how many types of secondary structures of proteins are basically present, right? After which so that helped us to understand the structure function relationship of the proteins and about the protein architecture. Followed by in week 3 we talked a lot of things about the protein folding, then denaturation. Okay, so protein folding, denaturation, and then uh, we took the example of a few proteins, and we wanted to discuss like when a protein folds or basically unfolds, right? So what are the different conditions at which a protein can act differently, things like that. After which there was like the protein isolation and characterization of how recombinant DNA technology plays a very important role over there. Though we discussed about the recombinant DNA technology much later than that, yet it was important to understand RDT at some point of time, and how we play around with proteins, different types of protein engineering, DNA engineering, recombinant DNA technology, the different DNA enzymes. Then there were few like the endopeptidases, exopeptidases, right? After which we wanted to talk about like the protein ligand interactions. Okay, so the protein ligand interactions, which were very important, and that like mm, included the different types of macromolecules or the different ligands that can be present in uh, interacting with the proteins. Then there were membrane proteins. Then a part of the proteins included the enzymes, the different types of enzymes and things like that. We had the metalloproteins, the motor proteins. So all the different types of proteins we talked about, and we also spoke like on the recombinant DNA technology and a special topics like the intrinsically disordered proteins, some viral proteins, things like that, right? And protein-protein interactions. So today, like we will talk a little bit about protein, maybe like protein macromolecule interaction. Okay, where we will have a few macromolecules, though we are mostly aware of the techniques and things like that, but yet we need to talk about these things. Um, this few things are there. And as you might be aware that there is an exam that is coming up very soon, which you all have to give very well, because yeah, so all this course or these live sessions were all about the doubt clearing and things like that. So we have to be like we had spent a lot of time like going through these weeks one by one and talking about these things in details about the proteins. We had covered a lot of portion on proteins. And so if you are ever into like the protein chemistry or in a protein lab, this course is a very interesting course, a very good course, I would say, and a course that one must always take if you are studying on proteins. So yeah, I would wait for like in during the last after I complete what I have to teach today, then I can wait for the doubt clearing if people have any doubts and things like that. So we can do that after we have completed it. And also, like after the chapter is done, there will be like a few assignment questions, like we always have on protein macromolecule interactions. So today's special live class session will mostly cover protein macromolecule interactions. So I'm sharing my slides with you all. So if my screen is visible to you all, I feel we should start. So the special topic is protein macromolecule interaction. So we will start with the protein carbohydrate interactions, that are intermolecular and intermolecular interactions between a protein and carbohydrate moieties. So carbohydrates are very important biopolymers and have variety of functions and serve as a recognition element. So most of them are specially recognized by other biomolecules. So proteins which bind carbohydrate structures are known as lectins. Compared to the study of protein-protein and protein-DNA interactions, it is relatively new and thing like that. So this interaction is basically a lot more novel, and people are studying a lot nowadays on these lectins and trying to uh, invest a lot of more time and energy in doing a lot of research on these things. Now, uh, okay, just okay. So many of these interactions that involve the carbohydrates are found at the cell surface as a part of the membrane glycoprotein or glycolipid. Now, these interactions can play a role in the cellular adhesion and other cellular recognition events. Intracellular carbohydrate protein interactions refers to those interactions between a glycan and polypeptide moieties in glycoproteins or the glycosylated proteins. Now, there is a classification where you have lectins. So, lectins are kinds of proteins that bind the carbohydrate recognition uh, domains or the CRDs. So, we could use different CRDs to classify them. One includes the C type, or the calcium is required to activate the binding. So, calcium binds to the protein and the carbohydrate by the non covalent bonds. And there are two types. So P type has two types of like the mannose 6 phosphate can recognize the phosphorylated saccharide. One is cation dependent and the other does not require a cation to be activated. So I type lectin is named from the immunoglobulin like domain. So cyanoadenosine is one of the I type lectin which binds specifically to sialic acid. Then you have the antibodies. So most antibodies, so we all know what antibodies do, and there is a lot of antibody engineering that is going on nowadays. So mostly antibodies have been similar structure except the hypervariable region, which is called the antigen binding site, and this region is constituted by the combination of various amino acids. So when the antigen is a kind of carbohydrate polysaccharide, the binding could be regarded as a protein carbohydrate interaction. Now, the biological function of these lectins or these sort of interactions play an important role, like cell adhesion, signal transduction, post pathogen recognition, inflammation, stabilization of the protein structure. Now, there are different methods for studying these. Most common one is the X-ray crystallography, like it gives you a detailed information on the interaction between the carbohydrate and the protein. NMR is possible, so if you do some kinds of titration, like the NOSI experiments or the nuclear overhauser effect spectroscopy (NOE) experiments, so that will give you a much better idea about the specificity, the affinity of binding, association constants, and the equilibrium thermodynamic parameters of this binding. Then there are others like the molecular modeling, molecular docking that will help you to dock these structures and help you understand the binding interactions, map the binding interface, the residues that are playing an important role, all of this within the protein, as well as you can look over those things in the carbohydrate through a uh, one dimensional NMR as well, or if you're solving a structure, then the complex can be solved to get the protein carbohydrate complex molecule. Fluorescence spectroscopy will also give you an idea, so it has advantages like no procedure of separation and plenty of ways to get the fluorophore source. So there are some amino acids and ligands that have fluorophore after they're activated. Then comes the polarization interferometry. So it's a label-free analytical technique for measuring the interactions and the conformational changes. So part of it includes the glycoconjugates. So glycoconjugates is general classification for carbohydrates as are covalently linked with other chemical species such as proteins, peptides, and other compounds. These are includes the categories of glycoproteins, peptides, and glycans. They're involved in cell-cell interactions, including the cell-cell recognition, in cell matrix interactions, in detoxification process. So you can see over here, this is the phospholipid bilayer. You have the glycoproteins that are visible on the top, the extracellular region. Then there are a few glycolipid carbohydrate interactions that are taking place with the phospholipid bilayer. Then outside the cell, you will see also these carbohydrate protein and carbohydrate lipid interactions both happening. So one is a glycoprotein, the other one is a glycolipid. So glycoproteins are the carbohydrates has an oligosaccharide chain, the glycan that is covalently bonded to the polypeptide side chain of a protein. So sugars in glycoproteins are attached either to the amide nitrogen atom of asparagine or to the oxygen atom of serine or threonine. Erythropoietin is a glycoprotein hormone present in blood serum that stimulates the production of RBC. So most blood group antigens are glycoproteins and the specificity is mostly determined by the oligosaccharides present on the surface of RBC. So glycoproteins includes the O-linked glycoproteins or the N-linked glycoproteins that varies in this particular carbon, where it is either linked to the oxygen or the nitrogen. The other portion can be basically the same, just there will be a change in structure as it's interacting with a different atom. Now, glycoprotein structure, so these are proteins that are covalently attached to the sugar residues. So the hydrophilic and the polar characteristics of the sugars it dramatically change the chemical characteristics of the protein to which they are basically attached. Now, the addition of sugars is often required for a glycoprotein to function properly and reach its ultimate destination in the cell or organism. Now, glycoproteins may serve many functions in the body. Some glycoproteins provide structure, example collagens. Some glycoproteins are involved in immunity, example immunoglobulins or immunoglobulin G. Mucins are secreted into mucus of the respiratory and digestive tracts, but the specific mucins can retain water, thus allowing mucus to serve as an effective lubricant. Specific glycoproteins present on the surface of red blood cells determine the blood group type as well. So basically, lectins are types of proteins that can bind to specific carbohydrates. They bind to soluble carbohydrates or to a carbohydrate moiety that is part of the glycoprotein or glycolipid. So lectins usually contains two or more binding sites for carbohydrate units. They typically agglutinate red blood cells, therefore lectins are also called hemagglutins. Some of the plant lectins are concanavalin A, ricin, abrin, wheat germ agglutinin, soybean lectin, and the peanut lectin. So the two important classes of lectins are the C type and the P type. The C type we discussed on the C type that requires a calcium ion for binding. So this calcium ion is required to link the sugar residue to the lectin. And the P type requires a phosphate group. Now lectin's functions is to facilitate cell-cell contact and communication. Beta carbohydrates on the surface of one cell binds to the binding sites of lectins on the surface of another cell. Binding results from numerous weak interactions which come together to form a strong attraction, and a lectin usually contains two or more binding sites for the carbohydrate units. Now, in addition, the carbohydrate binding specificity of a certain lectin is determined by the amino acid residues that bind to the carbohydrate. You see, it's required for a cell surface oligosaccharides, the lectins. So you have cell 1 and another cell, so binding of lectin to the particular oligosaccharide. Now there can be different like the C-type lectins, also known as the collectins, which are part of the innate immune system. Their function is to bind oligosaccharides on the surface of the microorganisms. Binding of collectins to microorganisms may trigger elimination of microorganisms by aggregation that complement activation, opsonization, or the activation of phagocytosis. Now, C-type lectins can also be the selectins that play a role in the immune system by recruitment of white blood cells into inflamed tissue. White blood cells normally do not interact with the endothelium of blood vessels. However, inflammation causes the expression of selectin on the surface of the blood vessel wall. Selective carbohydrate units on the surface of white blood cells present in flowing blood can interact with selectin that results in the rolling of the white blood cells on the endothelial cell surface, followed by stable adhesion and migration of the white blood cell into the inflamed tissue. So you see over here how a selectin uh, works. So the function Selectins are stored in seeds that function as the storage proteins. They also function in defense, where an invading microbe or insect disrupts the plant's cellular contents. The stored lectin that may agglutinate or sicken the pathogen or predator. Ricin is a toxic lectin that is present in the castor seeds, which inhibit protein synthesis at ribosomes. Now, legume lectins generally bind either glucose, mannose, or galactose, and they are involved in the attachment of nitrogen fixing bacteria to the legumes and in the protection of the pathogens. So, we had previously talked about IMSA, and this is a very important topic, though. So, right. Okay, so but we have not discussed about how to do it. We had talked about a little bit on IMSA, just a portion. We touched on IMSA. So IMSA is basically such a technique, you can say, that is very important when you're. So this particular technique is very important and a very powerful technique in detecting specific binding of nucleic acid protein complexes. Like 30 years, for the past 30 years, it has been the go-to assay to investigate the qualitative interactions between DNA, RNA, and nucleic acid binding proteins. Also, one can say that this is a technique that gives you an idea about a lot of things. Like you also get an idea about the association constant, the dissociation constants, and things like that. So it's very much similar to like a titration you do, and then like an ITC. So you titrate at different conditions, you add different concentrations of DNA, and you allow it to interact. You plot a graph, and you get it. So it's a rapid and sensitive method to detect the protein complex with the nucleic acids. So what is EMSA? It's not like a electrophoretic mobility shift assay, but it can also be known as a gel shift assay or a gel mobility shift assay, band shift assay or a gel retardation assay, based on how you are doing it. So it's a technique to detect whether protein is attached with the DNA or RNA or not. So as we know that many cellular processes where the protein needs to interact with the DNA or RNA, like in a transcription or translation. Now, originally utilized broadly in the investigation of sequences, specific DNA binding proteins such as transcription factors, IMSA have been additionally developed to explore DNA protein interactions, RNA protein interactions. IMSA is a sensitive method using radioisotopes to label nucleic acids and autoradiography. It is possible to use very low concentrations and small sample volumes. The principle behind EMSA is that you have a labeled DNA or a labeled RNA that can be any labeled, like uh, f- there can be a fluorophore that is labeled, or some kinds of an isotope labeling like a 32p. And then what you do, you titrate, you add an unlabeled protein, and you add a constant amount of DNA, and you vary the protein amount, and you run it in a native page. So the native page is important. You do not run SDS glycine or a tricine gel. We do not do all that sorts of things. It's a native gel because we want the complex to be intact, and that is what we observe under a fluorescence channel. So how it works? So the steps involved in the working of EMSA is the preparation of a cell protein extract, prepare radioactively labeled DNA, then there is a binding reaction, non-denaturing gel electrophoresis, then detection of the outcome. Now the protein binding to the unique site on the DNA will form a complex PD in equilibrium with the free components. Now when there is a strong interaction between the proteins, the DNA, then Ka greater than equal to Kd and the distinct band is observed. However, due to dissociation occurs during electrophoresis, faint smear might appear between the two bands, but that's absolutely fine. If single DNA molecule has multiple binding sites for an individual protein, there will be multiple complexes formed and we observe many bands. So we can understand that just from one experiment, we can get an idea about so many important things. So the protocol is that we have to make labeled DNA. So it can be a double-stranded DNA, a single-stranded DNA, or a single-stranded RNA, double-stranded RNA, hairpin DNA, or RNA, whatever it is. But we have to make it labeled. So either we fluorolabel it or we label it with P32. Whatsoever we do it, the labeling is important. Then comes the biotinylation, then the preparation of a gel. So we have to ensure that the gel plates, pressure, combs are all clean, and we need to remove the fingerprints, any kinds of DNA. So it's recommended that there is a fresh set of apparatus and gel components specially made for IMSA, for performing IMSA. Then we create the polymerization mixture and we put it to avoid any kinds of bubble formation. It's always recommended that we pre-run the gel. So pre-electrophoresis is recommended. So we remove the comb and we allow it to like run for 30 minutes. We equilibrate the gel with that buffer and then we load the sample. But before loading the sample, we have to make sure that we are cleaning the wells where we are going to load the protein, because these are very critical and sensitive experiments. Like even if there is a small amount of problem that happens in the gel, the entire thing gets spoiled and then we have to do it once again. So the sample preparation happens at minus at 0 degrees or in ice, where you need to keep the DNA or the RNA, because these are fluorolevel as well as very sensitive. At the same time, you have to do it in dark conditions such that there is not exposure to light, which can lead to the hampering of the fluorescence. Right, then we perform the electrophoresis, like run it at a constant voltage for like 40 minutes, 45 minutes, and then we take the gel and observe it under autoradiograph, where you see visualize the bands. But there it's the way I had said it, it looks very simple though, but it's not like that. You have to do it the entire thing in dark condition, in cold condition, and the buffers have to be fresh. You have to handle them very carefully, because after all, you're working with a nucleic acid that's very sensitive to degradation and things like that. Also there are protein degradation which needs to be controlled. Like there are certain proteins that degrade very fast, though most of the transcription factors are intrinsically disordered proteins only. But that is absolutely fine, because if you can purify them very well then and do these experiments quickly, it will avoid all sorts of problems. Now there can be multiple binding sites and multiple ligands can bind there, or there can be even multiple proteins binding to one ligand, or one 2 to 3 ligands binding to one protein. So you will see different changes in the shift, which we call as the shift. So you will see the change. So that is what we actually observe. So there can be multiple multimer verses that are forming, though like the image that we see over here is very clean. Like you have one band of DNA, it's like crisp bands. It usually does not happen in this manner, but you can understand that. uh, that is why it's very sensitive, and you have to do it clearly to understand them, and then you have to analyze them properly. So this is the similar manner. So you have the free probe, you have a particular shift, and then a super shift. From there you can understand like whether it's a competitor or non-competitor and things like that. So how and why this titration is done? So as we know that we're going to have a DNA interacting with a protein, and for that to happen, we need to have enough DNA and protein in the solution, and then you add. So this is the free DNA. So uh, you have the similar amount of DNA with an increasing amount of protein. So at the last lane you will see only most of the protein left, and in the first lane you will see most of the DNA left. And the point at which 50% of the DNA is utilized by the protein is the value of Kd, the predicted value of Kd, I would say, because the calculated one you will do after you plot a graph. So the application involves the study of conformational changes in DNA. For example, IMSA2D variant with electron microscopy is also used for conformational understanding or characterization, analysis of cellular extracts from the presence of certain DNA binding proteins. The bound and free DNA are separated using IMSA. It proves useful for stoichiometric analysis, a number of protein that binds DNA fragment. It is also used to study whether transcription factor is attached to the DNA or not, if the transcription factor is targeted or not on their activity. So the advantages includes that it is can be used with wide range of nucleic acid sizes, structure, as well as wide range of proteins. It's a relatively simple method to perform, used for qualitative analysis. It proves useful in detection of protein that binds to DNA and cause difficulty in migration of fragment on the shell. So highly sensitive method and is used for low cost, no special equipment is needed. So in this technique, it is possible to use both the crude protein extract and the protein recombinant proteins and the purified recombinant proteins. So although it is a very advantageous and sensitive technique, but it has a few drawbacks, like there are a lot of restrictions that we need to have when we are doing EMSA. It does not provide a straightforward measure of the weights of the proteins, as mobility is influenced by several other methods as well. It's not an appropriate method for kinetic studies. Dissociation is one of the biggest drawback of IMSA. It occurs during electrophoresis, thus prevents detection. Interacting biomolecules must have different electrophoretic mobilities. And in conclusion, it is used for various purposes such as quantifying interactions between DNA and proteins. It is widely used for the detection of protein-DNA interactions and works on the observation of protein-bounded DNA as compared to a free DNA. Any questions still here? Any questions? No? Okay. So we should discuss the practice problems. So which among the following is or are true about the proteoglycans? So the options that we see over here, um, proteoglycans are a class of macromolecules. that are found in the extracellular matrix. They consist of the core protein to which one or more glycosaminoglycan or GAG chains are covalently attached. If we look into the first statement that says they are present in both extracellular and intracellular matrices, that's incorrect because proteoglycans are a major component of the extracellular matrix. We do not find them in intracellular things. Statement 2 is correct. The core protein can have one or more gag chains attached, and these chains are long, unbranched, consisting of repeating disaccharide units, so they can be typically be fitted as well. Statement 3 is incorrect because this chain, gag chain, is connected to a serine residue on the core protein, but this linkage is formed via a specific tetrasaccharide bridge. No. So option 3 is correct. I feel. Option 3 is correct. And heparin sulfate and chondroitin sulfate are types of proteoglycans. That is incorrect because these are types of glycosaminoglycans. So 2 and 3 are correct. That is option B. The next one says oligosaccharide microarrays are used to determine. So oligosaccharide microarrays. So these are tools that are used to analyze the interactions between the carbohydrates and lectins. The microarray consists of a solid surface with various oligosaccharides attached to it. Now, when you take a solution of lectins and apply it on oligosaccharide, specific oligosaccharide, they recognize that and it helps you to detect which oligosaccharides are bound to lectins, and then you can get an idea about the specificity and affinity of the lectins towards the different carbohydrates. So therefore, the specificity and affinity of carbohydrate binding to lectins is the correct one. Glycosphingolipids are the plasma membrane components in which the dash head groups are dash. So glycosphingolipids are glycolipids that are found in the plasma membrane. They're composed of hydrophobic ceramide lipid tail and a hydrophobic head group. So the head group is a chain of sugar units, which are a type of oligosaccharide. These head groups are exposed to the outer surface of the cell membrane and plays a very important role in cell recognition and signaling. So therefore, the head groups are hydrophilic and are oligosaccharides. So that's option B. Which of the following structural motifs is generally not found in RNA binding proteins? So from the name, if you see, RNA recognition motif or RRM is present. Double-stranded RNA binding motif, that is also there. Even the zinc finger motif has a role to play in the RNA binding proteins. Helix-turn helix does not do that and is a common DNA binding motif, and it's not present in RNA binding proteins. So there is a test that is given, homoglobin sum test, to determine the modified form of hemoglobin. Which among the following statement is or false about a particular test? So glycated hemoglobin or is formed when glucose molecules non-enzymatically and irreversibly attach to hemoglobin. So statement 1 and 2 are true. The test measures the average plasma glucose and over a prolonged period, and is a form of that is chemically linked to sugar, but it is not formed through an enzymatic pathway. So 3 only is incorrect. So this question, the next question is basically you have to tell which types of lectins are this. So X is for cell addition, Y is for development and maintenance of nervous system, Z is for generation of functional lysosomes. So cell addition means it's a C type. Y is a P type because it's for the development and maintenance, and for generation of lysosomal functional lysosomes you have the I type. So CPI, that is option D. What is not true about RNA footprinting assay? So proteins that bind to RNA sequence can protect the region of the RNA from RNA's digestion. That is correct. The region that is not protected is known as the footprint of the protein. That is wrong. This makes it possible to locate a protein binding site on the particular RNA molecule. Correct. This method uses RNAs to cut the end labeled RNAs with or without bound proteins. That is also correct. So only option B is incorrect. Interactions of proteins with nucleic acids are essential for which of the following biological processes? Is it DNA replication and repair? Yes, it is. RNA processing translation, very important. Gene expression, absolutely. Regulation of chromatin structure, that is also important. You have the histones. So 1, 2, 3, and 4 all are correct. The next one says which among the following is not a function of replication protein A or RPA? So replication protein A is a single-stranded DNA binding protein that is essential for DNA replication, repair, recombination, and its functions includes protecting the single-stranded DNA from being degraded by the endonucleases, binding to DNA during the nucleotide excision repair process, and preventing single-stranded DNA from forming secondary structures or winding back on itself. But option D, it regulates the transcription of genetic information from DNA to messenger RNA, is incorrect. Okay. Now we have the syndicans and the glypicans. These are major families of membrane heparan sulfate proteoglycans. There are a few fill in the blanks which we have to fill up. Also, the next one is for the histones. So let us see how these things work. So syndicans are transmembrane proteins that are held by a plasma membrane by a hydrophilic interaction. They have a core protein that extends to the membrane and is attached to a sequence of polar amino acids. Whereas glypicans are attached to the plasma membrane by a covalent bond to the GPI anchor or the glycosyl phosphatidyl inositol, which is a type of a membrane lipid. So V is syndicans. So syndicans are held by hydrophilic interactions between a sequence of polar amino acid residues and plasma membrane lipids. However, glypicans are held covalently attached to the membrane lipids. And histones, so histones we know are proteins that have high content of positively charged amino acids, specifically lysine and arginine. The positive charges of these amino acids allow them to bind tightly to the backbone of DNA that's negatively charged, helping to pack the DNA into a chromatin. So the histones have a high content of lysine and arginine as well as positively charged amino acids. The next one is, which of the following methods is used for the detection of protein nucleic acid interactions? tRNA footprinting assay. So it's a method used to identify the binding sites of proteins on tRNA molecules, but it's not a general method for detecting protein nucleic acid interactions. But the others, that are pull down assay, chromatin emitter precipitation, IMSA, all can be done. So 1, 2, and 4. Okay. Now we have a big question. A DNA binding tetrameric protein with four identical subunits operates by a helix turn helix motif. It's DNA binding domain which inhibits the expression of genes coding for proteins involved in the metabolism of disaccharide in bacteria by reducing the transcription of the proteins. What is the name of the DNA binding protein? How does the bound repressor reduce the transcription of the specific protein? So basically, it's we can understand it's the function of a lac repressor protein. It functions by prompting DNA looping, obstructing the RNA polymerase binding site. So option D. Okay. Yeah, I am done. If you have any questions now, you can ask me. No questions then? Okay. Thank you very much. And so this is basically our last class, like I said before. So in so you guys have exams, an exam that you have the people who are taking it go through the lectures properly. These doubt clearing sessions will be very very helpful for you. There are so many questions that we discussed which have been asked in the previous years or are will be asked, so there might be repetition of questions or things like that, similar kinds of question or the exact questions. So go through these questions very, understand these questions clearly, and yeah, so that's all I had to say, and thank you very much for joining this 12 week uh, for joining for this 12 weeks on this course on fundamentals of protein chemistry. And that's all I had to say. Best of luck for your exams and for your future as well. So yeah, best wishes. Thank you.